个 Department of Molecular Cellular 的 Biology Biochemistry。那他的 PhD 呢是在 Harvard University 的 Department of Molecular Cellular 的 Biology。那他很特别的就是，他在PhD 毕业之后呢，在简正鼎中央研究院简正鼎老师那里做 postdoc 长达7年，所以我们发现今天是一个非常温馨的聚会哈，大家就是发现那个大家很多的共同点都是简老师。那我们今天就非常，简老师好像蛮老的。那我们因为大家都是简老师的 offspring。那今天非常荣幸，我想我就把时间交给 Margaret 啊，那由她来给我们这个精彩的演讲，大家给她一个 big hand。谢谢。
Okay, I want to thank Ya-wei and also Institute of Neuroscience for giving me this opportunity to visit and also present some of our recent work here today. So the title for my talk is glial cells in physiological and pathological brains. So I will divide my talk into 2 parts today. So the first part will be on a brief summary of some of my past work and also on ongoing unpublished story in the lab. And the second part will be on some of my future work. And because of the time constraints, I won't be able to show you all the data that we have, but I will show you some of the key ones in hope to convince you that these projects are worthwhile doing and also with great potential. So as you all know that in the brain, neurons are the major mediators for the brain function. So they form synapses and also they mediate synaptic transmissions. Glial cells, however, they are passive bystanders, and they are often passive, and also they are known to provide nutrition to the neurons, but they are not excitable. So which means that they have been thought to be passive regulators. So there are 3 major groups of glial cells in the central nervous system. So the first group is astrocytes. So they are known for their star-shaped like structure. The second one is oligodendrocyte. They are known to ensheath axons and provide insulation. And the third group is microglia cells. So they are smaller cells in the brain, and they are very efficient in engulfing the cell debris and also clean up the nervous system. So furthermore, they are very inflammatory, which means that they release pro and anti-inflammatory cytokines into the brain, and that will balance off the brain inflammation state. So before I go into the function of these cells, I want to let you know that we study glial cell in 2 different animal models. One is Drosophila, one is mice. So the reason why we are doing this is because these cells in both animal models, they are quite similar in terms of their morphology and their functions. For instance, in Drosophila, we have ensheathing glial cells, which can ensheath the axon very similarly to oligodendrocytes. And also, we have astrocyte-like glia and cortex glia. Both can function similarly as mammalian astrocytes. So we know that Drosophila glia can participate in a variety of cellular processes, such as neuron stem cell differentiation, synapse formation and activity, and also synaptic remodeling. So by studying these cells in 2 different types of animal models will allow us to understand better how this type of cells in the brain can function in different cellular contexts. So I want to give an overview on glial cell function here. So we know that starting from the very beginning, a precursor cell have stem cell-like potential, which can differentiate it into either a neuron cell or a glial cells. So once glial cell overproliferate, it can generate glioma, which is a type of very serious brain disease. We also know that one of the classical functions for glial cell is to provide extracellular cues to help with axon guidance. And we know that more and more recent work have shown that glial cell can mediate synaptic choices, neuronal remodeling, and circuit connections. With all this new function of glial cell in the synaptic context, we now know that glial cell can regulate a wide range of different physiological behavior. Now, furthermore, we also know that glial cell can participate in the injury response mechanism, and also glial cells are known to regulate neuron degeneration. So with all this diverse role, some of our work and also other colleagues' work have shown and identified the ubiquitin proteasome system, UPS, as a major degradation pathway in glial cell. So starting from my postdoc career, we have identified 3 different types of E3 ligase-related components that contribute to gliogenesis and also the development of a Drosophila neuronal structure called mushroom body. So this work has been published and also have tried to elucidate the glial function from a developmental perspective. Now, we also know that glial cell can participate in injury response mechanisms. So two of recent stories in my lab have shown that the lysosome hydrolase caspin-D, CAD-D, that can be translocated into the cytosol, where it's in a neutral pH environment. It can add as phosphatase to dephosphorylate cophylline, which then regulate actin dynamics, mitosis, and also microglia injury upon microglia migration upon injury. We also try to model fly with traumatic brain injuries. So basically what we did is we hit the fly head with a device, and that could cause potential brain injury. And then we purify out the glial cells, in this case extracells, and then we perform RNA-seq analysis. And using this approach, we were able to identify a newly activated pathway only in extracells that is mediated by a receptor called PVR and is JNK related. So all these findings have shown that glial cells participate in this injury response mechanism. So with all these previous findings, we then switch our attention to whether glial cell contribute to neuron degeneration and what is the mechanism of glial cell contribute to neuron degeneration. So before I start, I want to introduce a group of disease called neurodegenerative disease. So most of you have already known that this type of disease include Alzheimer's disease, Parkinson's disease, and also Huntington disease and ALS. So all these diseases have common features, and one of the common features is that they involve the degeneration of different types of neurons in different brain regions. And another common pathological hallmark is that they often seen, there's often protein aggregation and inclusions in neurons of in the brains of these disease patients. So, for example, in PD, we know that there are Lewy bodies, which are the name for the protein aggregates, that these protein aggregates are composed of alpha-synuclein. And in AD, we also know that there are protein aggregates that are composed of amyloid beta and also tau. So very interestingly, these protein aggregates are seen in neurons. But what's more interesting is that these protein aggregates have been shown to be detected in glial cells in patients such as multiple system atrophy and also in ALS. So this has raised great interest in my lab and some other labs that whether they are similar or distinct mechanisms that to eliminate those protein aggregates in neurons and glia, whether these protein machineries they serve as they add as a cell type specific manner, or they basically they are similar. So all the proteins are utilized in the similar ways. So to understand this will help to understand how to target these protein aggregates so that we can find a way, a therapeutical way to eliminate those protein aggregates, and then we can perhaps try to eliminate the disease progression. So we know that in neurons, autophagy is one of the major ways to clear alpha-synuclein in PD. So this is a brief recap of the autophagy pathway. So we know that autophagy pathway involves de novo synthesis of autophagosomes, which can then fuse with lysosome and then generate autolysosomes and for substrate degradation. There are a number of PD-rich genes that have been shown that mutation associated with these genes also exhibit autophagy defects. So the current hypothesis in neuron is that under homeostatic condition, autophagosome can be generated de novo at the distant axon terminals. And then once they are generated, they can be trafficked back to the soma area where there are abundant lysosomes, and they can fuse and become autolysosomes. And that's available for substrate degradation. But under pathogenic condition, however, the formation and also the trafficking of those autophagosomes are disrupted. So once they are disrupted, not enough autolysosomes are formed in the soma area for substrate degradation. Therefore, we are seeing substrate degradation is blocked, and we are seeing protein accumulation. So that's what happened in neurons. So then what happened in glial cells? Do glial cells also possess the same degradation machinery in eliminating the protein aggregates? So my lab has been focusing on trying to address these questions. And in the past couple of years, we have identified a new factor in glial cell that contributes to the alpha-synuclein clearance in PD. So I will tell you about the story in the next couple of slides. So to do that, we have to, we first set up a flight model for neuron degeneration, and we use Parkinson's disease as an example. So we know that first for PD patients, they exhibit several symptoms such as progressive loss of locomotor activity. They also have reduced lifespan and progressive loss of DA neurons. And also in their brain, we saw alpha-synuclein Lewy body aggregates. So in flies, what we can do, we can somehow mimic these symptoms. We call it PD-like symptoms. So for example, we can analyze the adult climbing ability in order to tell whether there's potential movement disability. So to do so, we construct a semi-automatic machine for analyzing the climbing distance for thousands of flies all at one time. So this is quite a high throughput manner. We can also analyze Drosophila lifespan, which perhaps can tell us about early human death. And we can also analyze DA neuron loss in the Drosophila brain. So there are conserved DA neuron clusters in Drosophila brain, which are very sensitive to alpha-synuclein, because people have shown that alpha-synuclein overexpression can cause those DA neuron loss at the particular cluster of this in the Drosophila brains. So with all this assay set up, we will be able to tell and associate with genetic manipulation whether there are certain gene or factors that can cause PD-like symptoms. Now, similarly, we can do so in mice as well. So we set up the footprint and paw analysis to try to get an idea about the movement disability of these mice. And we also set up the injection of this preformed alpha-synuclein fibers into the mouse brains. And this way, we can monitor the pathological forms of alpha-synuclein, and we can monitor their levels by immunostaining. And we can also analyze the DA neuron loss at a brain region called substantia nigra SN, which is a region that we know that DA neurons at these regions are particularly vulnerable to PD. So with all these tools set up at the beginning, back in 2017, we started initial behavior screens that using the adult climbing distance as a marker. So we identify in this screen a gene called GAK deoxin. So GAK stands for cyclin G associated kinase, and deoxin is the homolog of GAK in flies. So you can see that this gene, this protein contains multiple domains. It has kinase domain, P10, and also cadherin binding domain, and also a J domain. J domain is the domain that interacts with Hitchcock protein. So we know by this, we know that this GAK deoxin can act as a kinase, and also it can act as a cadherin encoding factor. So it has multiple functions. We also know that GAK deoxin expressed ubiquitously in both neurons and glial cells. And furthermore, there are previous literature suggests that GAK mutations are associated with clinical PD patients. And also GAK has been found to regulate alpha-synuclein expression level, although only in cell culture. So with all this information, we believe that we are in the right direction because we screen out a potential PD risk factor. So now what we want to do is to really study this gene and see whether it functions in glial cells to contribute to PD. So thanks to fly genetics, we were able to selectively inhibit gene expression in a special cell type. So that's what we did at the beginning. We inhibit D-axon expression in glial cells. And then we analyze different PD-like symptoms using the assays that we set up previously. So very interestingly that we saw when we inhibit glia, the axon expression, we see a decrease in the lifespan, suggesting that this fly died much earlier compared to the wild-type control. We also see that this fly climb much slower, so their movement, their locomotion is defected. And also, the defects in the locomotion is in an age-dependent manner, so it's progressive loss. Also in mice, we found that using footprint analysis and also paw tests, we were able to see that the walking behavior of these mice is impaired. And also by paw tests, the movement time that takes the mice to climb down from the up point to the down point is much longer compared to the control. So with all this result, we believe that we are looking at a glia factor that without it, in the absence of it, can cause locomotor deficits and shorten lifespan. Now, what about DA neuron degenerations? So again, we can utilize, study the Drosophila adult brain and also the DA neurons in the areas, SN area, to see whether this neuron degenerate. So this is exactly what we saw when we, in the absence of D-axon or microglia GAK, we were able to see that DA neuron degenerate in an age-dependent manner. So this kind of fit into our hypothesis that glia, GAK D-axon function in glial cell and might contribute to PD. So now, what about alpha-synuclein protein levels? So alpha-synuclein accumulation and aggregates is the common pathological hallmark. So the very first thing we did is to generate a binary system in flies. Because fly does not have endogenous alpha-synuclein, we have to overexpress it. So what we did is that we overexpress exogenous alpha-synuclein only in neurons, while we can manipulate gene expression such as D-axon in glial cells. So to really make sure that this system works, what we did is that we express alpha-synuclein in neuron first, and then we purify those glial cells in the adult brain. And we want to see whether there are traces of alpha-synuclein in glial cell. And by Western blot analysis, we were able to see some traces of alpha-synuclein in glial cell when we only express alpha-synuclein in neurons. This suggests that we are seeing a transmission of alpha-synuclein from neurons to glial cells because there's no endogenous alpha-synuclein in flies. Now, using this system, we were able to inhibit the axon expression in glial cells. And what happens is that once we inhibit the expression in glial cell, we see the overall significant increase of these alpha-synuclein levels in the brain. So this would suggest that glial axon help to mediate alpha-synuclein degradation. Otherwise, without it, then we see accumulation of alpha-synuclein levels in the brain. Now, similarly, in mice, we also see that doing brain slices and probe by immunostaining the pathological form of alpha-synuclein, we were able to see an increase of these pathological forms of alpha-synuclein in mice that lack microglia GAK. So this is also a consistent result with fly. And also, very interestingly, what we saw is that this accumulated alpha-synuclein co-localize much better with the components along the autophagy and also lysosome pathway. So the co-localization between alpha-synuclein and LC3, which is autophagy marker proteins, and also LAN1 is significantly greater compared to the control. So this result gives us a hint that the degradation of alpha-synuclein could be through the autophagy pathway or the lysosome-related pathway in glial cells. So to really test whether this GAKD axon function in glia is pathologically relevant, what we did is we again performed the alpha-synuclein preformed fiber PFF injection into the mouse brains. So these are preformed fibers. So once they are into the brains, they will form aggregates and they can propagate. So what happens is that we do observe this PFF in the control mice. But what's really interesting is that in the absence of microglia GAK, we were able to see a further enhancement of this PFF in the mouse brains. So that means that without glia GAK, there is a significant amount of alpha-synuclein accumulated, and they are in fiber form. So with all this behavior phenotypes, we now wonder mechanistically how does GAK deoxin mediate alpha-synuclein degradation in glial cells. Now, because of the result, preliminary results suggesting that this pathological forms of alpha-synuclein co-localized with LC3 or LAN1 better, we decide to look at lysosome first, because lysosomes are the terminal destination for degradation. So again, thanks to flight genetic, we were able to label this lysosome selectively only in glial cells. So what you are seeing here in greens, this is a model for the Drosophila adult brain that we analyzed. But what you are seeing here in greens are all glial lysosomes. So if you look at the top panel, when we inhibit deoxin expression only in glial cell, we see a dramatic increase of those glial lysosome signals. But if we inhibit the oxin expression in DA neurons, we were not able to see such increase. So this result suggests that we are seeing a lysosome defect specifically and only associated with glial cells. Now, if we look at it more closely, we see that these lysosomes, they are larger in size and also higher in number. So EM analysis also shows similar result that we are seeing these larger lysosomes and also a higher number of lysosomes in the brain. So this result together tell us that without glia deoxin, the lysosome biogenesis is affected. So we want to see whether the effect on lysosome biogenesis is conserved in mammals. So what we did is that we set up two systems. One is the microglia cell line called immortalized microglia cell called IMG. And we also manipulate GAK expression in this cell line. And what we saw is that in the absence of microglia GAK, similarly, we also saw lysosome aggregation. And in the brain slices that, of mice that lack microglia GAK, we also see increased LAN1 positive lysosome signals that indicates that there are potential lysosome aggregation, and also the biogenesis could be affected. So with all this data, we believe that we are looking at a conserved function of GAK and deoxin in glial cells. Now, in addition to lysosome biogenesis, what happened to lysosome function? Because lysosome function is more related to whether the substrate degradation can be successfully achieved. So we then decide to look at lysosome function. And one of the common ways to look at lysosome function is by probing the lysosome acidity with a tracker called lysotracker green. And this dye can stain leaf brains and help to understand, try to monitor the acidity of the lysosome. Lysosome has to be at the proper acidity in order to degrade substrate. So if there's something wrong by staining with the lysotracker, that means that these lysosomes are not properly functional and they could not degrade substrate properly. So by analyzing lysosome acidity using lysotracker in either in a single lysosome scale or in a global brain scale, what we saw is that indeed in the absence of oxalin, we see a decrease in the properly acidified lysosome number. So that means that we are without glia deoxin, this lysosome, they are not properly acidified. Now, if we try to analyze the function of the other two lysosome hydrolase, CAT-B and also CAT-D, what we found is that the CAT-B activity stand by this magic red dye is significantly decreased. Also, the processing of this CAT-D, another hydrolase, is impaired. That means not enough mature CAT-D available for substrate degradation. So all these results suggest that we are looking at glia deoxin function in glial cell that regulate lysosome acidification and also its activity in substrate degradation. So how exactly does deoxin do it? So we want to know in more details how deoxin regulate lysosome acidification. So the way we do it is by a fly rescue experiment. So I just want to make sure that people understand this part, because sometimes the rescue experiment is very hard to understand for non-fly person. So in flies, we can do genetic experiments to try to get rid of a gene expression. So in this case, we can do RNAi expression. So we express RNAi and no oxalin. So we are seeing, like previously I showed you, that the lysosome acidity is wrong, like it's not properly acidified. But what we can do is that in the background of this no oxalin background, we can put back deoxin. So this is what we call rescue. So we can reintroduce the gene expression to see whether we can rescue or restore the phenotype that we are monitoring, in this case, lysosome acidity. So indeed, when we put back deoxin expression, we were able to see that lysosome acidity is rescued. So this result suggests that we are really seeing a phenotype that is caused by deoxin. And when we put back deoxin, we're able to rescue it. Now, if we put back gene expression of the human GAK, which is the homolog for deoxin, we were able also to rescue this phenotype. So that means that we are seeing a conserved function for the fly deoxin with a human GAK. Now, very interestingly, when we put back a deoxin mutant, a variant that without the kinase domain, we call it deoxin delta kinase, we were not able to rescue this lysosome acidity defects. So this means that the deoxin kinase activity is very important, and without it, we were not able to restore the lysosome acidity, and it's probably involved in regulating this lysosome acidity in glial cell. So this is the statistic. So because of these hints and these results, the next thing that we did is to perform a phosphoproteomic analysis in trying to identify potential downstream phosphorylation substrates. So we got a lot of data and we tried to analyze it. But turns out that one of the proteins that caught our attention is VHA44. And its residue, serine 543, it's the residue phosphorylation. This residue phosphorylation is significantly reduced in the absence of deoxin, glial deoxin. So what is VHA44? So VHA44 is a subunit of a V1 complex and for this V-ATPase. We know that V-ATPase is a lysosome enzyme that it localizes on lysosome membrane. And it localizes on lysosome membrane, and it can be assembled by V1 and also V0 complex. Once it's assembled, it can pump the proton into the lysosome. And therefore, it can control the lysosome acidification via this way. So so now that we narrow down to this VHA44, that is one of the subunits for V1. And its phosphorylation at one particular residue is affected by deoxin. So we next performed co-IP analysis, and we found that indeed deoxin and VHA44, they form a complex together. And also, if we inhibit the VHA44 expression in glial cell, what we found is that the lysotracker staining is greatly reduced, suggesting that it affects lysosome acidity. So all this point to a role for VHA44 acting downstream of deoxin, that interact with deoxin acting downstream of oxalin in regulating this lysosome acidification. So to really support this hypothesis, we go back to our fly genetic experiments. So again, we show that without deoxin, we see a defect in the lysosome acidity. Okay. Now, if we put back this time not deoxin, but VHA44, then we were able to rescue this acidity of this lysosome. So this is a lysosome that co-stand with LAM1 and also lysotracker. That means that they are properly acidified because they can be stained and detected by lysotracker. But in the absence of deoxin, we were not able to detect as much of those lysosomes as the control. So now if we put back VHA44, we're able to rescue and restore this phenotype. But what about putting back a VHA44 mutant that has this serine 543 residue mutated to alanine. So this is a mutant forms of VHA44. So very interesting, when we block out the phosphorylation at this particular residue, we were not able to rescue the lysosome acidity in glial cells. So this is the statistic. So up to now, we believe that VHA44, this phosphorylation at this residue is an important event downstream of the oxalate, and it controls lysosome acidity. So next, I told you that VATPase requires the assembly of V1 and V0 assembly in order for it to properly function. So we want to know next whether the phosphorylation on this subunit will impact on the assembly of this VATPase. And whether this is the mechanisms that cause the lysosome acidification defects. So what we did is we tagged this two subunits, one in V1 and one in V0, with different fluorescent reporters. So one is with GFP and the other one with red RP. And we want to analyze the co-localization of these two different reporters in order to tell whether the VATPase is properly assembled. So we did a quick test in the cell culture. So we inhibit GAK expression by siRNA and analyze the colocalization of these two different fluorescent reporters. And what we found is that, indeed, that there's the colocalization of these two domains of V1 and V0 is significantly reduced, suggesting that in the absence of GAK, that VATPase assembly is impaired, and it could be the reason why that lysosome acidification is impaired. So with all this data, I would like to summarize with a model here. So I told you about how we identify glial-specific factors that without it, it causes PD-like symptoms. So without GAK deoxylin, the flies die early, and also they climb much slower, and also they have DNA neuron degeneration, also in mice. And the reason why we are seeing all this phenotype is because without deoxylin, the assembly of this VATPase on lysosome is impaired through this phosphorylation defects on this one of the subunit VHA44. And therefore, not enough proton is being pumped into the lysosome, and therefore this lysosome degradation is blocked, and we are seeing alpha-synuclein accumulation due to the block of substrate degradation. So that concludes the ongoing story in the lab that we are currently trying to wrap things up and trying to write it up. So since glia and neuron degeneration, this hot topic is one of the reasons focused on in my lab. So we didn't wonder whether there are other glia-mediated mechanisms contributing to neuron degeneration. So during the past couple of years, we do uncover some of other types of mechanisms. For example, for the first one, for the same factor, GAK deoxin, we have identified another function of it during autophagy initiation. So we found that GAK deoxin can also interact with a master regulator of autophagy initiation, ULK1, in the autophagy pathway. And by interacting with it, it activates autophagy and regulates autophagy initiation in glial cells. So this work is recently published. And we also identify another PD risk factor called LRRK2, DLRRK, that can regulate lysosome biogenesis in glia as well. And also, one of the ongoing story in the lab is that we identify a snare protein called VAN7, which originally we know that snare protein are functioning in a vesicle fusion. However, we uncover a new role for this snare protein in regulating mitochondria and lysosome contacts. So by regulating the contacts of MLC contacts in glial cell, it regulates the dynamics of mitochondria in glial cell, and therefore regulating the DNA neuron survival. So all this result contributes to our understanding of how glial cell regulate neuron degeneration. So that's all about my past and ongoing work. So now I want to tell you a little bit about our future work and what we want to do in the future. So as I told you that glial cells have diverse role in the nervous system, and since we have started working, trying to elucidate the mechanism of glial cell in neuron degeneration. One of the part of the focus in the future will be on continue to explore how glial cell contributes to neuron degeneration. And we also want to try to understand how glial cell regulate a type of physiological behavior called aversive behavior, which I'll tell you a little bit more later. So I subdivide my future direction into a couple of subpoints, and I'll tell you one by one, but due to the time constraint, I won't be able to go through this part, glia in depression related to PD. So if you are interested, please ask and we can discuss. So for the first part, we are interested in how glia, whether there are other types of glia-mediated mechanism of alpha-synuclein pathology in PD. So I told you that protein aggregation is the hallmark for neurodegenerative disease. So some of the key questions include how are the inclusion formed and eliminated in glial cells? But another key aspect of this is that how do the pathology, alpha-synuclein pathology spread from cell to cell in the brains, particularly maybe from neurons to glial cell. So you can see that this is a review figure telling you that how these different types of protein aggregate, they spread around the protein region, different brain regions, and they spread around as protein aggregates. So this is very interesting because no one has really studied how this pathology spread and whether the spread pattern depends on the cell types, whether there are distinct modes of spreading from neurons to neuron or neurons to glial cells. So we are very interested in this type of this question, and we would like to address it in the future. So one of the ways to address this is to take advantage of what we have already. So we already have setting up this genetic binary system for screening glial factors. So we can go back to it. And now we can still overexpress alpha-synuclein neurons and try to perform a screen in glial cell, try to inhibit different candidate gene expression in glial cell. And once we do that, set up the flies, we can assay for different types of behavior. For example, alpha-synuclein levels, locomotion, and also lifespan and neuron degeneration. So all these assays are already set up in the lab, and we can quickly get onto it. So for fly is a very good system to perform this high throughput screening. However, if we identify some novel glial factors, what we can do is we can move to the mouse system to study the mechanism more in depth. So for example, for this, the key aspect that I said that we are interested in, what we can do is we can again, took advantage of the previous setup system for the alpha-synuclein fiber injection. And then since these fibers are tagged with probe, what we can do is we can analyze this alpha-synuclein spread in the brain. And perhaps we can manipulate this novel glial factor that we found, manipulate the expression of those, and try to see whether it facilitate or inhibit the spread. And this is set up in collaboration with groups in US and China. And this has been set up in the lab, so we can also do it quite easily. So using these strategies that we can define the alpha-synuclein spread pattern from neurons to glia and perhaps look for new regulators on the spread. So I want to emphasize that the reason why we are interested in this spread pattern is because the way that they spread is very important for the disease progression. So if we can find a way and to understand better how they spread, we will be able to help to understand the disease better and perhaps to ameliorate the disease progression. So this is our ultimate goal, to really to find a way to perhaps cure the disease. So some of our preliminary data show that we do see evident alpha-synuclein fibers in microglia, as I show in our GAK story, and also in oligodendrocytes and astrocytes. So this preliminary result tells us that the system will work in terms of seeing alpha-synuclein fibers in different cell type, and perhaps we can analyze their spread pattern using different experimental methods, such as brain slices, fixed preparations, or volumetric staining using iDisco. So this is the first future direction. And for the second part is that we would like to search for a protective PD strategy by understanding the switch of microglia polarization. So we know that microglia, they can be polarized into two different states. That's the classical view of microglia. So you can see here that people have shown that microglia can be M1, which means that they are pro-inflammatory. They release like different cytokines. And it can be M2 as well. That means that they release different anti-inflammatory cytokines that makes the brain anti-inflammatory. So as I told you earlier on, that microglia are inflammatory cells. So this is a very simple way to classify microglia by M1 and M2. But in reality, this is not the case. So during PD disease progression, so there are multiple types of microglia that will have been detected. For example, they can be amyloid with enlarged cell bodies, or they can be stress-prime, dark, or dystrophic. So all this type of microglia have been shown in the PD disease patient or PD model mice. So this makes things much more complicated, and it's not very easy to understand how this microglia, how they mediate, manage the switch of these different types of morphology. However, it has been hypothesized that during PD, at the very beginning, we know that microglia cells can turn into M1. It's actually, they release pro-inflammatory factors, so that actually make the disease worse. But then toward the end of PD, we know that microglia can turn into M2, which is anti-inflammatory cells. They release anti-inflammatory cytokines. So if we follow this hypothesis, then it's very important to understand how a microglia cell can turn from M1 to M2. Or not M1 to M2, just any type of morphology, any type of state that microglia are switching. So we want to understand how they switch. So like, was there a key genes or key mechanisms that can regulate their switch so that we can perhaps artificially or manually or in control to try to switch them so that we can perhaps help with the disease progression. So to do so, we start out with a very simple approach. So basically, we took advantage of our cell lines, microglia cell lines, and we can use pharmaceutical agents to polarize it into M1 or M2. And then once we do that, we can do RNA-seq analysis and try to find key genes that correlate with their polarization states. And then we can verify this gene's expression in mouse PD models. So that's what we did, and we do get some preliminary data. So as you can see here, we performed the RNA-seq analysis, and we found some key genes in LPS-induced microglia. So, and because we have a lot of information, so what we did is we performed some screening through to make sure that we get what we want. So the very first level of screening, what we did is we do geo-annotation of these differentially expressed transcription factors. Why do we decide to look at transcription factor? Because we would imagine that if microglia want to switch from M1 to M2, let's say, it has to be achieved by a global change of gene expression, because that would be easier. And that can be done by transcription factor. So that's why we focus on transcription factor at the very beginning. So we got some potential transcription factor that are affected by LPS. And now we cross-compare those genes with a single-cell RNA-seq analysis database in human striatal glia cells in PD. This was recently published in Nature Aging. So we compared these two databases, ours and theirs, and we found that this one potential transcription factor called CEBPP, CEBPD, its expression is upregulated in both our microglia and also their striatal glia cells. So this gene is a potential transcription factor that are already known to involve in immune response. And its expression, it can work as heterodimers. So you can see that these are two different genes, but they work as heterodimers. And its expression is upregulated in our LPS-induced microglia cell and also in this database. And very interestingly, what we found is that when we induce the microglia with IL-4 to turn it into M2, we see this gene is reversely downregulated. So we believe that we are seeing a gene like a potential transcription factor that its expression correlate with microglia M1 and M2 polarization. So we believe that this might be a target for controlling the M1 M2 polarization. So to really test its disease relevance, so we quickly set up MPTP-induced PD mouse models. So we know that this is not the most perfect PD model because it's drug-induced and it's not transgenic. But for our purpose, we will be able to test this gene's expression and its disease relevance using this model. So what we did is that we set up two different models. One is acute and one is chronic, using different timeline of injection of MPTP. In the acute model, we inject the MPTP every 2 hours in 1 day. And in the chronic model, we inject MPTP in every 1 or 2 days throughout the 7 days. And what we saw is that in both models, we see very severe DA neuron degeneration, suggesting that this model is successfully established. And then very interestingly, what we saw is that when we analyzed microglia in these two models, we saw that in the acute model, this microglia, they have enlarged cell bodies, and also their process links is much shorter, significantly shorter compared to the control. So this means that this microglia, they are more like M1 state. But whereas in chronic stage, when we analyze this microglia, they also have the enlarged cell bodies, but their process link is not really affected. So I think we are seeing two different types of microglia here that they might be distinct, and one might be more into M1, and the other type is more into M2. So by analyzing acute and chronic models, and we also show we also try to analyze this CBBB, CBBD expression in these two models. And very interestingly, correlating with our cell culture data, we do see that in the chronic model, where it's more M2 state, that we see these two genes expression is downregulated. Whereas in the acute model, we do see only one gene, CBBD expression is upregulated. So for this part, we will continue to dissect the mechanisms of this CBBB and CBPD in microglia polarization in the PD context. So finally, I want to tell you a little bit about our preliminary work on glia in this type of behavior called aversive behavior. So because we have been working with this type of cell for so long, so we know that this type of cell is very important for almost all aspects of brain function. So in addition to neuron degeneration, we decided to look at other types of brain function or behavior. So for flies, it's actually quite easy to do because we can set up different assays in flies to really test their behavior. So one of the behavior that we start with is aversive taste memory. So let me show you how we did this. So we basically tether one single fly on a cover slip. And this single fly, because it has a taste receptor in both their mouth and also on their leg. So we supply fructose. This is a sweet taste compound onto its leg. So basically, the flies receive the sweet taste signals, and it knows that, oh, this is something they like, because people and flies, they always like sweet stuff. So once they receive the sweet taste signals, it will extend its mouth by proboscis extension. We call it as a PER response, which we can monitor. So then after the fructose, we supply the fructose, we then supply a bitter taste compound called quinine onto the mouth. So at this point, the flies can associate the fructose and also the quinine taste. So once they associate it with, so this finished the conditioning. And once we supply again the fructose, it will remember that if they got the fructose taste, it will follow with the bitter taste compound. And then they will not extend their mouth because they dislike the bitter taste. So then if that's the case for normal flies, we won't be able to see any PER response because that's what we quantify. And very interestingly, when we perform this type of test, when we block out glial cell activity in flies, we see that this fly actually like the bitter taste compound. They approach it and they do not avoid it. So we believe that this is part of the initial observation that we believe that glial cells in flies can actually regulate, is involved in this type of aversive or avoidance behavior. So because we know this at the very beginning, then the next thing is that we are trying to see how this type of behavior is regulated in flies, and whether there are responsible neurons that can work together, interact with glial cell to regulate this type of behavior. So we turn our attention to this type of neurons in the Drosophila mushroom body, which is the L-shaped LIGO structure in the Drosophila. And we know that this Drosophila mushroom body is the center for associative learning. And you regulate a wide range of behavior like long-term memory, sleep, and locomotion. And so we decided to test the neurons in MB and see whether they also regulate this type of behavior. And very interestingly, there's a type of alpha-beta neuron in MB that also, when we block their activity, we see that there's an increased response for the fructose, which means that they forgot about the aversive taste already. They don't remember it because this alpha-beta neuron or glial cell, their activity is blocked. So this type of alpha-beta neurons, they have been studied in many different cellular contexts, but we know that it releases acetylcholine. This has been shown to release acetylcholine. So the next thing is that we want to test whether glial cells do interact with this type of neurons. And we use a strategy that is called synaptic graphs, which allow us to reconstitute the GFP signals for two different cell types that they are nearby to each other. So that's what we did, and we found that indeed those glial cells interact with these alpha-beta neurons. And what happened is that since this is ACH dependent, when we block out the ACH receptor, nicotinic ACH receptor on glial cells selectively, we see a dramatic reduction in this reconstituted GFP signal, suggesting that ACH-mediated glial transmission occurs between alpha-beta neurons and glial cell, and this is mediated by ACH. So glial cells respond to ACH release from alpha-beta neurons. So I don't have time to go through more data, but I just want to summarize here that we know this is where we are right now. Alpha-beta neuron release ACH, and normally it would impact on following postsynaptic neuron we call the NBON and regulate ATM. But alternatively, we have identified a pathway potentially that might react to ACH and also release some sort of glial transmitters that will impact on the postsynaptic neurons and regulate this ATM. So this is what we hypothesize. So for the type of glial transmitter that glial cell release, we have some preliminary data suggesting that it could be ATP. So we will continue to explore this part and try to see whether the ACH-mediated ATP synthesis or release is the true mechanism for this type of behavior. So those results are all in flies. So what about invertebrates? Do glial cells also participate in aversive behavior in vertebrates? So this is something that was raised after we saw what we saw in the flies. And we decided to look at perhaps there's a particle in mice as well that can model the aversive taste memory. So we found one, and this is called conditioned taste aversion, CTA. So it's a very similar particle, as you can see, that we can supply the mice with a saccharine solution first, which is sweet. And then we can inject the mice with lithium chloride, which causes stomach ache. So it feels sickness. It's very uncomfortable. Once we do the association and pairing up, we supply again the mice with saccharine solution, and which will then, in normal mice, which will then probably the mice will reject it because it will associate with the lithium chloride already. So we call this a conditioned taste aversion and CTA mice. So we perform this protocol, and we test whether it was successfully established by quantifying the aversion index. So in fact, we were able to model the mice with CTA because we see a significant increase of aversion index, which means that they drink much more water than saccharine solution. So that means we got CTA mice. And then once we got these mice, we decided to look at their brains and see what happened in their brains. So we performed CFOS staining and throughout the brain regions and try to find reactive cells. CFOS stands for positive reactive neurons over glial cells. So we do a co-staining with S100 beta, which is an extracyte-specific marker. And what we found is that, very interesting, there's a group of extracytes in the brain region called lateral parabrachial nucleus, LPB, that is being activated. So we see that CFOS positive number is increased. So that's quite exciting for us because we then wonder whether we are seeing similar function of this LPB exercise in aversive behavior in invertebrates as well. So let me try to tell you a little bit more about LPB first. So this LPB region is in a brain region called PBN. So PBN region has been shown to be involved in the circuit that receives signal from upstream NTS and also propagate signal to different downstream regions. And it has already been shown that this region is involved in aversive behavior and feeding behavior. So I think we are looking at the right direction because this region is already known involved in aversive behavior. However, the extracytes in this region have not been shown to be involved in any type of behavior. So we continue to study and explore this type of LPB extracyte. And what we did is we use virus to infect LPB extracyte and activate it. And what we found is that in wild-type mice, we use two different approaches. In wild-type mice, we inject the virus in LPB to activate it. And we also use a Cree mice that is specific to extracyte, and also in combination with a DO virus to try to activate LPB extracyte. In both methods, approaches, what we found is that, very interestingly, these mice, they have a great reduction in the food and water intake, which means that they do not eat or drink. So this actually prevents us from assaying the CTA because they do not drink. So we won't be able to test whether they are avoiding the lithium chloride or the saccharine solution because they basically, this is more like an innate behavior. They do not drink or eat. So we are kind of stuck here because we don't know why, like we don't know how to test it, you know, like whether this type of mice still stuck with aversive behavior. But one of the things that came up is that whether this type of signals that are representing by LPB extracyte activation, whether it's these mice, they do not eat or drink, whether it's because that they are full already. They already have a satiety signal, or they just don't like the food. They just dislike it. So this is different. One is more on the natural requirement of the body, and the other one is more on like they just don't like it, not that they are full. So we try to perform another assay to see if we can distinguish these two types of signal. So the other type of assay is condition place aversion. This time, we do not use the taste as the signal anymore. We use the place, the spatial. We provide a spatial signal for the aversion. So this time, we put the mice in two different boxes. One is a pair stripe box with a pair stripe box wall, and the other one is a solid color wall. And during the conditioning time, we pair the pair stripe wall box with a CNO injection, which means that we activate LPB extracyte only in this box when the mice is in this box, but never in the solid color wall. So basically, we let the mice associate the LPB extracyte activation or the CNO injection with only the pair stripe. So this is called a conditioned place aversion. So very interestingly, when we perform this assay, what we found is that also by activating LPB extracyte, the mice was never in almost like very significantly reduced amount of time in this pair stripe box. They prefer to be in the solid color box. So we believe that we are looking at a signal aversive signal, not limited to taste, but also a spatial signal that is aversive, representing by LPB extracyte activation. So furthermore, we do a quick analysis on the potential involvement of upstream and downstream brain regions in this circuit. So very quickly to go through this, we activate LPB extracyte, and what we found is downstream PSTN regions is activated by, shown by C4 staining. And when we inject the mice with lithium chloride, we also see NTS and PSTN regions were C4 reactive. So together, basically, we believe that we are looking at a circuit that transducing the aversive signal from upstream MTS to the PBN, where the LPB extracyte is responding to the signal and then can help and interact with the neurons to transduce the signal downstream to PSTN. So this is where we are right now for this part. So combining with the fly part, so we believe now that extracyte function in aversive behavior is conserved in flies and mice. So there are a lot more questions to ask. How do fly extracyte regulate aversive taste memory? So we will continue to test the ACH-mediated glia ATP release to see whether this is the true mechanism. And how do mice LPB extracyte regulate aversive behavior? We will continue to identify more in details the LPB-related circuit pathway. Ooh. It's okay. And also the type of glia transmissions that, for example, what type of glia transmitters that glia cell release to impact on neurons, and the neurons, how they interact with glia cells. So I finished, sorry, too long. So I want to acknowledge the people that done the work. So I'm very fortunate to have a great team. So they work on two different directions, one on neurodegeneration and behavior. And all the reagent providers and collaborators, flight community, stock center, and co-facility, and a group of collaborators in China, USA, and Taiwan, and the fundings. And thank you very much. I'll take any questions.
那在这里，我在进入 Q&A 之前，我想跟大家报告一件事哦，就是大家最近可能会收到，就是之前收到说，哎，我们怎么 CV 一直在换？ 好，不是因为我们这个行政单位办事不力，而是因为好事发生。那个 Margaret 呢，最近就是 PNS 被接受，好发表，还有嘞，他也刚获得2030的国际年轻学者奖。
好，谢谢，谢谢，谢谢。
来，我们进入 Q&A。
Hi.
Thank you so much for the really.
Hi.
Thank you so much. It's really enjoyed it. For me, it's a very different world, but I have naive questions about the first part where you talk about the role of glia in neurodegeneration. So I think you elegantly showed that astrocyte lysosome is helping the degradation of these protein aggregates. So I wonder, so you kind of alluded in the end, so I wonder how do, at the cellular level, how do these proteins go from neurons to glia, and also when you manipulate when you demonstrate that these glia cells, so when they're doing their thing, cleaning up these protein aggregates in glia, does it affect the neurons? So do the neurons still degenerate as usual, and then so I'm guessing the question I'm asking is the phenotype that we're seeing here, is it because glias are important in general, and so when glias are not able to degrade the protein aggregates, then it leads to bad outcomes? Or alternatively, is it because they're helping the neurons to kind of fight up these aggregates? I don't know if that is making sense.
Okay. Okay, for the first question is on how they transmit, right? So there are receptor-mediated trafficking between these two different cell types. For example, alpha-synuclein, they are corresponding microglia receptor that can actually uptake alpha-synuclein on microglia cell membrane. So that's one way. Also, there are exosomes. So we know that neurons can release exosomes that can probably transmit. The other part is whether it's passive or active, right? Yeah, it's more like that. So we don't know, basically. So this could be two models could, work simultaneously. But my model, like the way I thought about it, is that because glial cell process can extend very long. So glial cell processes are all over, and they can wrap the neuronal soma and also axon everywhere. So it's probably more readily available when there's something like at the terminal, axon terminals, that those protein aggregates had to degrade it or whatever. So glial cell processes are right there, so they can actually update it and degrade it. So it's kind of a helping way, yeah. But whether if there's a defect in the like the degradation in glial cells, whether it causes DNA neuron degeneration, I'm not sure if it's the consequence or the cause of the consequence. Let's say, yeah. Yeah.
I'm sorry. Actually, I have a related question or follow-up question. Is that when you in the first part, when you knock down that GAK in the glial cells, I would assume that it will affect pretty much all kinds of neurons, although you say that you produce PD-like symptoms. But do you think that in that particular model, you actually have a more broad consequence of when you're knocking down GAK? And because some of the, like the lifespan phenotype that you show up really short. So I would presume that they actually have a much broader effect than just PD-like symptom. Can you comment on that?
I agree, actually. So for example, if we want to culture the GAK deficient primary microglia, we won't be able to do so because it's a cyclin G associate kinase, so it actually regulates cell cycle. So it is very possible that it affects other types of neuron degeneration, like their death as well. So I'm pretty sure there's a broader effect, but we haven't assessed those yet, so I'm not able to say, Okay.
So
所以我用中文问啊，所以第一个还是就是 lysosome pathway 我会觉得是一个非常 broad、非常 general 的 pathway。所以 Ao-Lin 在 neuron 有表现吗？还是它是 glial specific？ 因为你现在都用 repo driver 去 drive， 那当然就是在 glial。那可是它事实上它的 expression pattern 是 very specific in glial cell or just it's in all type of cells. So again, it's about the specificity because you claim that it's PD using PD model, but just lysosome and the protein degradation is much a general pathway, and it's probably affect many things.
Yes, that's a concern, but it's ubiquitously expressed. So it's also expressed in neurons and glial cells. So what we did is that we actually knocked down axon expression in neurons, but we didn't see any lysosome phenotype, and we didn't see any autophagy phenotype. So I think it still affects, so the neuronal expression of GAK or the axon is still very important, but it might not, the mechanism is not autophagy or lysosome dependent. So that's.
And my second question is about the aversive behavior. I noticed that you use like the temperature sensitive Shih-Chieh in glial cell to express it in glial cell, but and later you would suggest that it's probably a ATP release. If that's why you use Shih-Chieh, because Shih-Chieh is more related to like vesicle or membrane trafficking. However, I'm thinking that if ATP, generally I think ATP probably doesn't release from vesicle. It may be just from like in glia, in mammal, probably like pannexin type of. Those are maybe not Shih-Chieh dependent. So how do you explain that? How does the ATP affected by Shih-Chieh?
Okay. We use Shih-Chieh just to block the activity. I didn't really think about ATP at that point because we did that experiment quite long ago, like earlier on. But the reason why we, so let me see if that, I wasn't able to show you. Sorry. 这个我没有办法看到。Hold on, let me see. Here, the reason why we think it's ATP is because of this. I didn't have time to show you. So we purify the glia cell from the adult brains, and we do the brain incubation with ACH, acetylcholine, ex vivo. And then we analyze the gene expression of this glia cell that affected by ACH. And what we found is that there's an ATP synthase subunit that its expression is greatly enhanced. So we believe that, and also if we knock out the expression of this NACHR in glia cell, this enhancement is blocked. So we think that is ATP. Yeah.
So how does Shih-Chieh affect ATP release? Doesn't connect.
Right. So Shih-Chieh is physical endocytosis, right?
Right.
Right. So yeah, I don't know. Yeah. I can't say right now because it might not be. I'm not sure about in flies whether ATP release mechanism has been shown, but yeah. But we do know that in mammal glia cells that the ATP can be released via lysosome, lysosome exocytosis or other ways. Yeah. Yeah. Thank you.
I have two rather specific questions. The first one is regarding the first part. So when you manipulate the GAK expression, and it will affect the lysosome acidification, right?
Yes.
And when you overexpress the VHA44, it was able to kind of rescue the effect.
Yes.
Right. So I was wondering because so you were expressing the wild type VHA44, right?
Yes.
Not the phosphorylation mimic. VHA44.
We did both. Yeah, we did both. Yeah. So wild type was able to rescue, but the VHA44 mutant was not able to rescue.
But the mutant, but not the phosphorylation mimic one.
We have made the phosphomimic one, but we haven't tested yet. Okay. So it's 2E, right? Glutamine. Yeah, we haven't tested yet.
And for that, the way you manipulate the GAK was based on the RNA interference as well.
Yes.
Because I was curious, it's downstream, right? And it requires like kinase activity.
Yes.
And it's a bit interesting how can you overexpress the substrate in abundance, but it was able to rescue without the kinase to really phosphorylate the. Right. So was that suggesting there's another kinase that function redundantly?
Yeah, it's possible. Yes, I understand your point now. So you're saying like no more kinase, but you have more substrate.
For the aesthetic analysis, like when you regulate the gene expression, for example, right? So the downstream overexpression will be able to bypass the requirement of the upstream.
Right.
But in this particular case, the kinase activity is actually required to activate the VHA44 and for the assembly of the ATPase. So I was curious.
Yeah. So it could be that there might be other kinase or wild type VHA44 might work too. The phosphorylation.
Without being phosphorylated.
No, I don't know, but yeah, that's a problem. Yeah, we haven't. Yeah.
I think trying the phosphorylated mimic one may be an interesting.
You mean over phosphorylated, right?
Right.
Yeah. Okay. Yeah. We are trying it right now.
Okay.
Yeah.
My second question is about the aversion
behavior.
So because you went through pretty quickly, so the way to activate the glia was mediated by the G protein coupled receptors.
Yeah. You mean in mice, right? In mice. Okay, yeah. So we use a chemogenetic approach.
So for the chemogenetic approach, what will be the outcome for the activation? So the overall electrical activity is
the calcium. It's a calcium. So glia calcium activity is increased.
So it's possible that the overall, maybe the local activity will be increased upon the activation of the glia?
You mean like local?
The neuronal activity.
No, it's specific because the promoter is specific to extracyte. So we are only activating extracytes and only.
What will be the usual outcome for activating the glia cells? Are they gonna just activate the neurons as well?
It could be because they interact with neurons too. So once we activate the astrocyte, they can propagate the calcium wave. They do not transduce action potential, but they can propagate the calcium wave to other astrocytes or affect nearby neurons. This is possible, yes.
So it would be an alternative idea to think maybe the local activity is altered upon the activation of glia, right?
Yes.
It doesn't need to be just the glia network somehow later release some specific stuff to affect neurons. It could be the overall whole activity is altered.
Yeah, but the idea is that because glia are activated and they can release some sort of glia transmitter to affect nearby neurons. That's the normal, like more the widely accepted model that we have for glia-neuron interaction. So if it's just by, so calcium wave increase can cause release of glia transmitter. So this is the idea right now, like how they influence nearby neurons. That's what we thought.
So did you guys look at, for example, like a neural activity markers nearby to see whether there's some alteration in
we should do that. Yeah, that's what we are doing right now. Thank you.
So I have a similar question, it's a follow-up question. But before I ask you a question, I want to say, yes, I'm really very impressed with your talk, and you really can take questions from flymen and mouse scientists. It's very it's very unique.
Thank you.
You can, yeah, I think you know both systems. So I just wonder in the future, maybe during lunch we can discuss how do you run these two systems side by side.
Okay.
But I'm very impressed.
Thank you.
Great.
Thank you.
So now I want to ask the follow-up question is, so you use this C-force system to map this associative aversive memory, this kind of aversive test, or you say this is, you also use the chemogenetic activation of this LPB astrocyte.
Yes.
And they can recognize this place. This is aversive place test. So this is a kind of contextual memory, right? So you use the C-force to map this LPB. But I'm curious about in addition to LPB area, because this is aversive memory. This involve I think is many
other regions.
So do you only found LPB? Because I'm a little bit surprised that only you found increased C-force in LPB, because there are still other upstream and downstream. And also we know a lot of brain areas are responsible for this kind of aversive learning. For example, like this PVA and all other this maybe NAC, maybe BNST, maybe hippocampus, something like this. Can you comment on this?
I believe that we found other regions as well, but because our focus was on exercise, so I think the students really only look at the S100 beta and C-force co-localization. So, but I think, I'm not sure what other types of brain regions that we identify, but this is the most sensitive for exercise. That's why we follow up with this. Yeah.
This also means that this LPB circuit is important for this aversive memory.
Yes,
right?
yes.
Maybe it's but we know this LPB is in the brain stem, right?
Yes.
Area. So, but you show, in addition to this conditioned test, aversion test, you also show this conditioned place aversion test. Actually, these are a kind of conditioning associative higher cortical function. So there is a learning and memory plasticity. So in addition to this region, maybe this region is just the first station. They receive input, and this maybe just is the US US is the unconditional stimulation, and you have the CS, US-CS. So I believe there are more brain regions, higher cortical brain regions, responsible for this kind of aversive memory. And that's why I think I want to follow Ming-Chia's question. Maybe when you do this kind of manipulation, you activate LPB this is exercise, right? And you can just do a simple C-force mapping to check, to compare chemogenetic CNO control and also HM3 DQ, and then you do the subtraction. You do C-force for both.
I see.
You do subtraction. You will find maybe there are some brain regions the C-force increase,
Oh, okay.
but in other brain regions the C-force level is decreased.
Okay.
Okay. Also, I'm also interested in neuronal activity because if you activate the exercise, you increase the glial transmission.
Yes.
So what kind of glial transmitter and what's the target synapse? What kind of neurons are influenced pre and post? And also, where is the location of this somatic plasticity takes place, right?
Yeah.
So a lot of interesting questions.
Yeah.
If you want to study aversive memory, I think there are a lot of circuit questions you can ask, upstream, downstream, and we also want to know if it's aversive, so it's a reward aversive, and whether this dopaminergic system is involved. So if you are a circuit scientist, you show this brain region is important for this complicated behavior. We always want to ask you upstream and downstream circuit. So there are, I think it's a bunch of questions.
Yes.
Yeah. So that's
Yes.
why I said in the
Yes.
future you need to think in Taiwan, probably we don't have so many students. So how to run these two systems side by side is really a grand challenge.
Yes, yes. Thank you, thank you. Yes. So as you can tell, I start from flies to mice, so there's still a lot to learn in the circle level. Yes. Yeah. Okay.
I have been out of the scientific world for a few years, but I'm still enjoy your talk very much, and so fascinating still about how science, how basic science can tell us. So I first like to congratulate on your performance.
Oh, thank you.
接下来我要用中文讲比较好。
都可以。
就是你前面当然是做的，就是对于那个就有关于第一个部分哈，对于有关于这个 GAK 的这个部分。那刚刚前面几位老师都有问了说，它其实可能不是那么 specific for PD 哈。那我就很好奇，就是说你有没有探讨或者了解一下跟其他的neurodegenerative diseases 的关系，或者像是 amyloid 的 plaque 啊，就是它对于其他的 aggregates 的这个效果会是什么，或者是说有没有想过从clinical 的 data 有没有相关的 gene 受影响而影响了这个 degeneration 的这个过程？
所以好，所以就是GAK 的 mutation 它有 SMP mutations associated with PD 这样子，就已经有被发现过，但是跟其他种的这个 neurodegenerative disease 好像没有关系，就是在 GWAS study 里面是没有看到。对，那至于它，其实我的想法是，它应该是一个比较 general 的 regulator， 因为它其实 regulate cell cycle， 然后它也 regulate 这种 Catherine uncoding 的一个过程。那 Catherine uncoding 其实是很 required for protein trafficking、physical trafficking， 所以它可能的确这个 effect 是很多。那我们当然我们只是局限在看 PD， 然后只是去 analyze PD-like symptom 这样子，所以我 agree， 就是说它可能会有其他的功能，但是对其他种类的这种 neurodegenerative disease， 我想 so far 还没有很明显的证据说明它会有影响这样子。
你的那个实验用的是把 Shih-Chieh 的 fibril 打到脑子里嘛？哈，对。那没有想说打
其他的吗？
可能用
其他的吗？
对，其他的这个 mutant 的 mice 来。
对，可以，这些其实都可以做。如果有人有钱的话，都可以做。就是有的时候就是必须要先对 prioritize. Yeah. 谢谢。
好，那时间的关系，我们在这里结束今天这个精彩的演讲。那请大家给 Margaret 一个，
好，谢谢，谢谢，谢谢。